I always like to I'll start with a brief disclosure that I serve on an advisory board for Prelude Therapeutics, but that won't interfere with anything I'm discussing with today. I'd like to start with a quiz from the audience, and I can't see your screens, but this is kind of the thought experiment that I like to start with that motivates our research. So if you were to put yourself in my shoes and think which two pieces of information would be the most helpful if you were formulating a plan for a patient you were hoping to treat. And so on the left, you might know the age, the smoking status, and the stage or the degree of advancedness of a patient's cancer, or you just get the picture on the right. For by and large, I'd say most people will choose A. That's certainly what I would choose. The knowing a bit about the cancer and about the patient would help me a lot more at this point than knowing just this picture of their cancer itself. Well, what if I changed the scenario a little bit and instead gave you the information on the left, and I also gave you a little bit of molecular information, some of the things that aren't part of standard of care, but that the patient was had a certain mutation, likely responded to one class of drugs. But for this, I charged you 4 weeks, and your patient had to pay $6,000 or so. In this case, I think things get a little bit more questionable. And indeed, I'd still take the information just on the left because I wouldn't, it was going to delay my patient's care and not actually change treatment with the information I've included so far as part of current standard of practice. But if I changed the amount of information that was available to us, if I included, in addition to the response to certain types of drugs and mutations, if I included the presence of certain viruses which inform our treatment of head and neck cancer, or included additional components of risk factors to the patient and didn't have cost or time constraints, then I would much rather have the information on the right on B than on A. And what we are setting ourselves out to do, the goal we're hoping to accomplish, is that if we include that basic information that I've provided for you first, plus information about the pattern of cancer growth through the lens of artificial intelligence called deep learning, that we can combine those two initial columns and potentially capture all of this information in the second column instantaneously and very inexpensively to improve the care for our patients. So the central hypothesis that governs this component of my work is that the patterns of cancer cell growth contains hidden information about all sorts of different biological signals. So these would be mutations you were born with, called germline mutations, mutations that your cancer developed over time, called somatic mutations, different patterns of genetic expression or activity that are happening within the cancer cells, presence of virus in the cancer, late stage modifications to the cancer programming called epigenetics, and then factors like response to treatment, recurrence risk, or other clinical features which can be identified rapidly, accurately, and inexpensively with machine learning techniques. So this is what we're going to discuss over the course of this evening. So what is this component that we're talking about? And when I say artificial intelligence, or when I say deep learning, I'm talking about the same thing, and that is a series of related mathematical functions which allows us to flexibly process digital imaging information. And there's a couple of basic schema here. Effectively, what we're doing is taking a graphic like what you see here as a house, and then on the right, we are reducing this graphic down to the most basic imaging components. So you might imagine just the red pixels on that graphic, and we are looking at 3 by 3 squares, and each of those 3 by 3 squares is passed through a matrix, a grid of mathematical functions, which reduces that 3 by 3 square down to a single square in this layer above. And through a series of really intensive computational exercises, we pass the same image up and down a series of these mathematical mathematical layers, which adaptively allow the weightings that are applied to each layer above and below to change over different iterations of this algorithm. So what we can do is effectively train a computer to see the components of an image which are most influential to predicting one outcome or another. So that's expressed here on the bottom in this layer of panels, of sheets, as it were, fed by an output on one end, or whether or not a sample represents cancer, for example, and all the way on the other end might be the pathology slide. So this is the technical term for this concept is a deep convolutional neural network, and we sometimes call it deep learning for short. And this is the same type of artificial intelligence which underlies things like Google image search. So at different layers of this network, just another example of what's happening, at different layers of this deep convolutional neural network, the system is learning different things. So at the very most basic layers, the layers right next to the image, the network effectively learns how to see. It sees very basic image structures like edges, like fill, like border. And these can be combined into more complex and specific image components. In this case, we're seeing eyebrows, we're seeing noses, we're seeing mouths, which can be subsequently combined through that same series of sheets of layers into something that's very recognizable. In this case, it's men and women's faces. And these are images extracted from different layers of a deep convolutional neural network trained to recognize men or women from faces on the internet. So this same concept can be applied to a lot of different categorization tasks. In other words, the same set of those basic components here expressed on the bottom could be combined to make faces, could be combined to make cars, elephants, chairs. And in our case, we can combine those same imaging structures to make cells in tumors. So why are we hearing? You probably have heard about this in the press and in academia. This is an area that is absolutely exploding: deep learning or artificial intelligence, and particularly in cancer. And so why is this happening? Why is this happening right now? Well, number one is there's huge amounts of publicly accessible cancer data that we have been able to use to train our models. And that's the Cancer Genome Atlas, which is housed in part here at University of Chicago. There are new methods, such as the deep learning method I just described, which makes analysis of this tremendously rich data set much more efficient and more highly performing. We have new types of computer hardware which make this analysis accomplishable. This was something that was the amount of computations that are required to run even the simplest deep convolutional neural network are things that were well out of our reach 10 years ago. And with the explosion of innovation in this realm on the hardware side, it makes it accomplishable for me for labs like mine with ideas, but not unlimited budgets, to be able to kick off our own in-house hardware and network processing. So I have workstations in my lab funded by the Cancer Research Foundation and others, which allow me to do this research on a reasonable scale as a junior faculty member. So what we've seen is just an explosive amount of academic productivity. You can see that this is an extremely sharp curve, which is the explosion of the number of articles in the past few years in deep learning in cancer. So this pipeline, I talked about this, using these simple visual features to make different kinds of accomplish different sorts of goals. And in my lab, that goal is applied to cancer cells. So what the basics of the pipeline is that we take a standard diagnostic slide that is ubiquitously available for patients as part of the workup for a patient as they are diagnosed with their cancer. We make a digital image of that glass slide, and then we identify the area that is of most interest to us. In most cases, that's the area of cancer on that glass slide or on that digital slide, and we have a team of great collaborators in the pathology department that help us do that. We then chop up digitally that area of tumor into much smaller and digestible parts. These we call digital tiles. They're of fixed size and can be fed efficiently and evaluated using those subsequent sheets, down to the 3x3 pixel, in our artificial intelligence specific hardware in our lab. And from that, we get a wealth of information, not just in terms of whether or not the artificial intelligence can identify a topic of interest, but also why it's identifying a topic of interest. And we'll talk about that. But we can think about the omics of this visual data, the high dimensional, all of the different nuances that the machine is using to identify a feature of interest. So in my lab, we've downloaded every publicly available digital image from the Cancer Genome Atlas, and we sought to see if we could identify the most frequent mutations in those digital pathology slides without doing an expensive and time-consuming genetic sequencing test. So we thought of this as a digital pathology next generation sequencing. So cancer patients, sometimes to identify a target for a given type of treatment, we would evaluate using a molecular test whether or not they have one mutation or another. And we sought to see if we could do this directly from the pathology slide, directly from the architecture of the tumor, in all of the most frequent types of cancer, in all of the most frequent targetable concepts. So we used our deep learning algorithm to identify every highly relevant and actionable genetic alteration in more than 4,000 patients from the Cancer Genome Atlas. And what we saw is that in many cases, we were able to identify many targets. So the metric we use to determine if we've accomplished this or not is a measure called the area under the ROC curve, or AUROC. This is a number that varies from 0 to 1, but in the middle, it means at 0.5, it means that you're no better than chance. And at 1.0, it means that you have perfect discrimination for the binary event you're looking for. So if any of these, as they get closer to 1, it means we are able to detect that genetic alteration with high degrees of accuracy. So we were quite excited about the fact that we were able to accomplish this in some cases, and clearly it's not all cases. But what we saw was the opportunity to democratize precision oncology. In other words, if you could, using just a simply stained slide, understand the genetic profile of a patient's cancer, we might be able to deliver precision oncology care much faster, much less expensively, and to a much broader geographic area than using our current sequencing-based platforms. So all of this, the other really exciting part of this for us as we were doing it, is that we came up with this idea on March 21st, 2019. I remember the brainstorm session. It was with my good friend Jacob Cather, who's a physician in Germany. And all of the work was done on the concept within 8 months, and we posted our preprint 8 months later. So this is the rapidity, this is the speed of research, which doesn't lend itself that well to the traditional cycle of grant funding, and which is why I'm so thankful for the alternative funding mechanisms like the Cancer Research Foundation that I've been able to avail myself of, because it's how we do really rapid and timely research. what we anticipate, even though the performance is not where we want yet, what we anticipate is that as more data comes into the system, the amount of performance, the quality of the digital biomarkers that we're producing will continue to improve. So in an experiment we did and published in a series of papers, we were able to detect the molecular hypermutated state of certain GI cancers called microsatellite instability using our artificial intelligence platform. And that what we saw over time was that the performance of our algorithm, the ability of us to detect this clinically actionable state in cancer patients, improved as the data set size increased. And in fact, in an additional experiment, we allowed the same set of validation or double checking individuals stayed stable, and we varied the amount of data available to train the model. And we found that, as is probably obvious, more data leads to better model predictions. So this reinforced our optimism that over time, we will be able to improve on some of these first pass concepts that we were able to show in our digital NGS concept. So there have been, I showed the explosion of concepts. And so this extends across a variety of different domains. I showed you some data that we produced on the mutation prediction, and other groups have come up with other important clinical biomarkers of response. So predicting survival across a number of different cancer types and predicting response to certain therapeutic interventions. So at this point, there's no FDA-approved deep learning-based digital pathology biomarkers, but there have been a number that have been shown to be valid in external concepts. So part of our exploration was, what are some of the other biological signals that we could discover using the same platform? And so one, something that we were really curious about is whether or not we could determine the ancestry or the ethnicity of an individual based on their pattern of their tumor alone. And so this is some preliminary data that we had from some time ago, and you'll see that same AUROC on the left hand column. And what we discovered was that, at least for a week, we thought that we had determined a dramatic difference between African Americans and Caucasians in terms of the way their tumor grows, and similarly among Asian Americans compared to Caucasians. So we were stunned initially at the degree of the degree of response in these tumors. So then we looked a little bit more, and what we discovered actually was that maybe not quite as exciting, but potentially even more relevant, is that what we were actually detecting was the differential proportion of certain individuals based on the place their tissue came from. In other words, you can see here, and that University of Chicago was by far one of the most frequent donators of African American samples to the Cancer Genome Atlas. And so, in fact, what we had determined was that in thinking we were detecting a signature intrinsic to African Americans, what we were actually detecting was a signature intrinsic to the University of Chicago, and even potentially more fear-inducing is that there were many of these, not just ethnicity, but many different important traits like stage of cancer, like progression-free survival, like HER2 subtype, which were differentially divided among institutions in some of these big cancer data sets. So we went back and thought a little bit about this potential for bias, particularly among the subset of models that were run for that progression-free survival outcome, and did not have an external validation. And just to comment that these are some superb research groups. This is Google that has been looking at this this outcome, this progression, this survival prediction algorithm, including pathology slides, which may be in some cases related to the pattern of tumor staining that's intrinsic to a place rather than to biology of an individual. So we defined as a result of this this what we call site-specific digital pathology signature, which is composed of the processing and components of how a place processes their sample, how it stains the sample, and how it digitally captures that image in the microscope, as well as the intrinsic population differences that are reflected in the individuals serviced by a given place. So we have proposed, this is work by Dr. Fred Howard, who's in my lab, and has proposed a way to get around this by altering the way we allow certain sites to be accessed by the training algorithm in order to reduce this site-specific digital bias and all of the implicit wrong suggestions we might be making as a result of it. So just to summarize the impact of this, on the x-axis here is the decrease in the area under the ROC curve with this bias reduction plan in place compared to a non-corrected model. And what you will see, I'll call attention to this tail of the curve up here. You may or may not recognize, but this is a combination of genetic signifiers such as ALK and PI3 kinase receptor, and also ethnicity statuses, which means that there is a dramatic decrease in the true prediction of these classes if we appropriately correct for this imbalance and bias. In other words, that many models might be susceptible to making dramatic, inappropriate inference about patient characteristics, particularly as it pertains to racial minority groups. And so I think this is incredibly important and impactful work that will allow us to understand the challenges within the data and responsibly and inclusively perform artificial intelligence research across a lot of different types of Americans. So what I think another theme that is reinforced in this work is this concept of accountability. So in other words, partly what clued us into this is that we started to see some differences between the pathology in different groups that didn't seem to be biologically motivated. And so as a practicing clinician, thinking about making a decision on my patient's behalf based on an algorithm, it gets really scary if it's not based on the right things, meaning if it's based on what clinic door you happen to walk in instead of what biology of your cancer actually is. And so we think about this concept of explainability in artificial intelligence, which effectively is, can we trust the results of an algorithm enough to make important clinical decisions for patient care? And the classic cautionary tale here is a machine learning algorithm that was trained to identify wolves versus husky dogs. So in some days past, authors downloaded internet pictures of wolves and internet pictures of husky dogs and trained an algorithm that was using similar methodology to what I've talked about, that was able to really accurately discriminate the two classes. And so when they went back in and tried to ask the algorithm, why have you made the decision you've made? What they determined was it wasn't looking at the animal's bone structure or the shape of its tail. It was actually looking at whether the animal was sitting in the woods or sitting in your living room. And so this is the kind of bias where a wolf only sits in the woods and a dog sits in your living room or in your backyard, which is highly accurate, but would be woefully inadequate for making patient care decisions if the analogy was carried forward. So we've thought about deploying the same sort of concept of explainability in our cancer pathology models. And so we have been working in this emerging subfield of artificial intelligence called generating adversarial networks, which allows us to synthetically generate a representation of what the network is seeing or what the network thinks is important. And so from the microsatellite instability or the hypermutation state example I gave before, we generated a series of images that represented either microsatellite stable or microsatellite unstable, right? And we compared them to the real images. And so I'm showing those here. You can see that this is the synthetic images, which are on the right. The pathologist was able to determine that they bore a lot of characteristic pathological patterns, and that you could find those same correspondent patterns on the real images, which was really exciting for us. And I think will allow us the opportunity to double check our work, make sure there's not a visible visual bias showing up in a generated network representation of a biological instance. And so as our networks become kind of more and more forward looking, we can make sure that we are indeed identifying things we think we are. So our current basis is that with a diagnostic digital pathology slide, we can run a network and we might train any number of different outcomes: treatment response, recurrence risk. I talked about genetic factors, maybe some other other model-based, scientific model-based responses. But I think in the future, what we really, really are excited about from a concept perspective is that the network can incorporate, and we hope will improve if we include additional types of data. So in addition to the diagnostic hematoxylin and eosin slides, maybe we can include stains for immunotherapy response. Maybe we can include stains for HPV status. Maybe we include radiology or clinical data. Maybe we can include even more advanced omics to further characterize and holistically include information about a patient's cancer state in order to make better decisions about their treatment. So I'm passionate about this, and I think that we're at a juncture where it's really important to support AI research in academic settings. There's a lot of great work that's being done by industry groups, but the reality is that the best questions are asked by clinicians, and the best clinicians are in academic practices, in my opinion. So some of the main things that we can that promote this specific growth domain is that domain-specific knowledge in medicine exists, is really important to ask the right questions in the right way. That large amounts of data is available in academic centers. We have the computer resources that are necessary to do these broad strokes concept research projects, and we have ready access to novel algorithmic development as part of our computer science group affiliate computer science groups. So I hope that University of Chicago is one of the places where we can be doing that research. I've been incredibly fortunate to be a beneficiary of CRF funding, and I wanted to, as I was reviewing or preparing for this talk, I also noted that many of my direct collaborators have also been beneficiaries of this. And so I wanted to just point out that there's really an ecosystem of quantitative scientists being developed here at University of Chicago, all driven by Cancer Research Foundation funding. And so Randy Swice and I are now co-PIs on the University of Chicago Cancer Systems Biology team science grant called the U01, which stemmed from my 2017 grant from the CRF. Sam Reisenfeld and I have a CDAC grant as co-PIs. which is now active, and I know that CRF is a funder of that, and Xavier Koitgen and I have a pending NIH grant as part of this same initiative. And then I share students or active research projects with a number of other investigators who are CRF funded at points before or after my time as a CRF YA. So this has been a potent tool to help build out our collaborative research. So I'm thankful. I've been very lucky to have support to be able to think innovatively and to be able to build some of these quantitative concepts. And I have a tremendous team that I work with, both here at University of Chicago in the Department of the section of Hematology Oncology, and with collaborators at DKFZ, with Bob Grossman's lab at Argonne, and in the Pathology department. So with that, I will conclude, and I will show my graphic of machines fighting the evil crab, which I think represents the way we think about our role here. So with that, I will conclude and see what questions are available.
So I have a question with regards to how you keep doubling back to make sure that issues like being really good at identifying where slides came from, as opposed to how do you keep testing that system?
So one of the ways we have, I think that one of the ways that we've done it in the past is looking really carefully and systematically at the extremes and at the population of guesses, what the algorithm actually thinks is important. And we've developed some ways to project the landscape of, even all the actual tiles that were pulled from cancer patient populations. We can look at them on top of, look at the tiles themselves, or look at the graphics of the cancer on top of the landscape of the genomics or the landscape of the AI model. And if you see, one of the things that I think we started seeing, particularly in our ancestry question, was that there seemed to be some persistent staining abnormalities that seemed, that when we got down into it, we were like, that looks like more than just a difference in the nuclei within the cancer. That looks more like more like something systematic or a bias. But I think that this is one of the things that you learn, and that I learned with my PhD back in the day, was that sometimes it just is really worth going into the raw data and checking cells of your spreadsheet, right? If the numbers don't make sense, then there's probably something wrong. And so effectively, that's what we're doing, is that we call it the hard, boring work of data science, which is just the evaluation, the grind through, making sure you really trust every step of your data from start to finish, and that nothing looks too good to be true. No errors are too low to be trustworthy. You expect that things are going to work less well than you hope, maybe, and that it takes a while to get to the results you'd like.
So we've got a question do you see the Q&A? I can say it. How can we collectively prevent introducing bias into algorithms?
Yeah, it's such an important question right now, right? There have been innumerable examples in the press about algorithms that are impactful to every component of our daily life, being found to have really insidious biases. And I think one of the main problems that is responsible for this is that oftentimes the data that we train an algorithm on is different than the data we use an algorithm on, right? And that's basically unavoidable. But the most important way to do that, to use algorithm in our algorithms in our daily life responsibly, is to make sure that the use case is as close to the training case as we can. So in the example I gave, I think it would be imperative. It's important to understand that if you were going to develop a tool based on our AI biomarker, you wouldn't be able to use it on African American patients outside of Chicago, right? Because it's too much of the algorithmics evaluation was tied to the treatment that they received at just one place. So in other words, it's not a generalizable algorithm. So going back to the question, I think the best thing to do is to gather as much data as possible, right? We should double down on responsible data collection, and do so acknowledging that there are biases boiled into everything, and there are opportunities to develop additional methods to help reduce the amount of bias in a concept, in a data set, in order to build the algorithmic applications that are most broadly available to use and benefit in society.
Dr. Pearson, is there an issue as you compile data sets and then they multiply as you create, pseudo data sets to check against them, about keeping all of this data, keeping it straight? I assume it's de-identified, but it could be could you talk a little bit about where we're headed in terms of issues with data sets?
Yeah, it's a great question, and I think that there's been a significant amount of thinking in the field about how do we. I think there's some people that no longer view data as de-identifiable, particularly if it's something like imaging data, where your tumor could effectively be a barcode for you as a person. Whether it was just one digital example or your tumor, period. So I think there's a question about the degree of privacy that can be guaranteed on the one hand, but on the other hand, there are also methods being developed to try and be as responsible as possible for data transfer. And so for example, there's a set of researchers who have developed algorithms that allow effectively you to keep the data on your own computer and then train just a little bit of the neural network, of a shared neural network, and then kind of do the same for a number of different sites. So one of the things we're excited about it from a research perspective is that this would be a great way to, if it worked and if there weren't other problems that we haven't found on them, this would be a great way to do international research, right? Because every country has its own privacy laws, but if a person's personal health information never left their home country, we might be able to produce really nicely generalizable, broadly applicable algorithms from data from every continent without having to sacrifice the privacy aspect or break laws that are vary tremendously from country to country. I think in our lab, we have tried to set everything up to be HIPAA compliant so that when data comes in, it doesn't leave. It sits in a secure server that the University of Chicago helps us administrate, so to make sure that it's not just me thinking about what could go wrong, but rather a lot of smart people thinking about the security of that precious data. But I think that's the kind of minimum buy-in, and we've realized from any number of different stories that there is no such thing as is totally secure data, right? Unless it's truly locked in a drawer. So it's a balancing act, and I think that it will be really, I think the methodological innovation will allow us to have to share less, which will be the ultimate best way to secure data in these collaborative research concepts.
Any other questions? I don't think we've got any, and we're right on time. So thank you. Thank you, Dr. Pearson, very much for your presentation and your important work.
Thank you. Thank you. I very much appreciate the support and the opportunity to speak with you all this evening. It's a topic I'm very excited about, so I'm very glad to share.
And thank you everyone for joining us, and we hope to see you in person very, very soon. The next time, or maybe the next time after that, but we'll be in touch to let you know, and hope everybody is well until then. Have a great evening.